This is a 3-mark question. We have to explain what is PCR and how it is helping to amplify small amount of DNA into multiple copies. So full form of PCR, you must learn that is polymerase chain reaction, which will be amplifying the DNA segments through a cyclic process of in which we have 3 steps. The first one is denaturation, that is heating to separate DNA strands. Then annealing, this is the cooling will be happen to add primers or short nucleotides which will help to elongate the DNA or the primer or the starter. Then extension, which will raise the temperature for DNA polymerase to synthesize new strands, and this will be extending in a way that primer will be extending to complete that DNA, double stranded DNA. In this way, this cycle will be repeated for 25 to 40 times in a machine called the thermal cycler. It will create billions of copies of DNA. So you have to learn the 3 steps, full form, and the name thermocycle. Let's figure it out. What are the requirements for PCR? The first one is template DNA. We need that template from which we have to make a millions or billions of copies, and that original DNA will be there, primers will be there. Primer is actually a small nucleotide which will be added to the template, and after that the elongation or extension will happen. This is the starter that we give. And then comes DNA polymerase. This is not a normal DNA polymerase which is present in other organisms. We are extracting it from Thermus aquaticus. So its name is Taq polymerase. This is because we are using a high temperature which will denature the protein. To avoid that, we are taking the DNA polymerase from a organism which has the capacity to withstand in high amount of heat. So that Taq polymerase is the enzyme. Then obviously it is nucleotides, that is the building blocks A, T, C, G, that should be there. That should be added like bricks to continue the process. Subscribe for more important questions for your biology board exam.